Hi, everyone. Welcome back to this is your second class in the psychopharmacology series. In this course, we're going to be starting to talk about some basic ideas in cell communication, both between cells and inside of cells. This is some very basic psychopharmacology concepts which will be used across the class when we talk about different drug groups. So this is a two-part lecture. This first part, I'm going to be talking about two of these 10 neurotransmitters. The remaining ones will come in the second part, and we'll talk about starting to understand how these neurotransmission systems work and some basic ideas in pharmacology, such as receptor affinity, excitatory and inhibitory constants, etc. So usually when I go to a conference, invariably there is some slide that looks like this. And my reaction almost invariably is, oh gosh, what is this? What am I looking at? And so I just thought I would start off with something a little simpler. And so I found this. Yeah, that's not much better, is it? So this is a close-up of that schematic of the brain. Don't worry, this is not what we're going to be using today. I just thought this was an amusing effort to attempt to graphically describe something that's very complicated, which is the human central nervous system. And as you remember from my suggestion last week, that we really want to think about learning as this sort of ascendant process, where each time you go over this material, and you will go over it many times in the course of learning it and in the course of your career, you're going to go a little bit higher each time. You're going to see the bigger picture, and you're going to start to connect the dots, which is a really satisfying feeling. So don't get discouraged if you don't understand it all the first time. I certainly didn't, and I don't expect you to either. You might need to listen to this talk a couple of times. So last time we talked about pharmacokinetics, which is how the drug enters, moves around, and leaves the body. So that's really what does the body do to the drug? And now we're going to turn our attention to pharmacodynamics, which is what does the drug actually do when it gets to its receptor targets? Or what does it do to other targets, which it's not necessarily intended for? The short answer is that it causes side effects. So this is really understanding what the drug does to the body. And I wanted to start off. I had my son help me illustrate these slides. He's studying art right now, and did a great job of laying this out for me. We were talking about how should we convey this information, and he suggested basically an organizational chart, like for a company, which I thought was great. So, welcome to the Brain LLC, and in this org chart, we're going to start off with the HR hiring and training department, which is really your genetic coding, your genotype. And from those genetic instructions are going to come a whole bunch of neurotransmitters, which we can think of as really the workers of this company. So we have the floor managers, GABA and glutamate, which we'll spend a lot of time talking about today. And then all these other neurotransmitters. We've got histamine, we've got melatonin, orexin, dopamine, acetylcholine, endogenous opioids, serotonin, norepinephrine, and endocannabinoids. So all of those are your different workers. They do different things in the nervous system, and some of them work together. So, for example, when we talk about drugs that are used to treat psychosis, we're going to talk about this important work group of serotonin and dopamine. Orexin and glutamate work together with certain drugs to promote wakefulness. And the experience of cannabinoids is modulated by glutamate, anandamide, dopamine, and GABA. So you can start to see how these groups work together. And sometimes they have these sort of network functions. So we won't get too much into this today, but these are a couple of areas of brain tissue that are implicated in issues around depression, for example. Your ability to concentrate on a task is largely regulated by the interchange between your default mode network, your executive, and your salience networks. So, without getting into too much detail about that, just understanding that there are these larger work groups, larger areas of the brain which may work together to create a function or may be implicated in dysfunction. Regardless, the end product is what we would think of as the phenotype. This is the output of this company, if you will. What is the final expression of these of all this work and all these work groups. We can think of this as the output or the phenotype. Sometimes we talk about the genotype, which is the genetic instructions, and then the phenotype is the actual organism. Sometimes we talk about diseases in psychiatry as disease phenotypes. So that's really, what does the disease look like? So somebody who has a low mood and is tearful and anhedonic and disrupted appetite and sleep, we might say that person has a depressed phenotype. So starting off at the top, HR and genetics. HR and training comes from our genes. And the genes are really a set of instructions that tell the body how to build proteins. Now, pretty much most of the things that we're talking about in discussion of psychiatry ultimately boils down to proteins. Receptors are proteins, neurotransmitters are proteins, and inside of neurons are additional structures that are made from proteins. So these protein instructions are really important because mutations of them may result in diseases. Now, back in the 90s, when I first started getting interested in psychiatry, there was we were calling it the decade of the brain, and this was this great hope that we would be able to fairly quickly untangle the causes of mental illness, and that we'd be able to trace them back to their genetic roots. Because if you remember, at the same time, the Human Genome Project was underway. And unfortunately, it turned out that there was not a single gene for, say, schizophrenia. There were many genes that were associated with schizophrenia, but then again, some people had schizophrenia and they didn't have many of those genes. So what it became clear was that the picture was much more complicated than we initially thought. And that you may have heard that there's genomic testing, pharmacogenomic testing, that we sometimes use in psychiatry. And what this tells us is whether or not a drug is going to be metabolized at a regular rate, at a slow rate, or a fast rate. It doesn't tell us whether or not a drug is going to treat somebody. Unfortunately, the way these tests are sometimes marketed creates an implication that they're going to that this will predict the outcome. What they really do is they predict whether or not somebody can tolerate a medication. In fact, we did a big study at the VA a couple of years ago where we looked at, we gave everyone genetic tests before they were treated for depression. And we looked to see, was there any improvement in the outcome of the group of people that were tested with pharmacogenomic tests to see, did that predict a better outcome for their depression treatment rather. And it turns out that it didn't. What it did predict was that people that were given the pharmacogenomic testing were were less likely to be treated with meds that would be intolerable. But unfortunately, both groups turned out to have about the same outcome. The group that was prospectively pharmacogenomically tested and the group that was not. So that has some benefit, but not a whole lot, and certainly probably isn't useful in widespread population treatment. We shouldn't be, it's probably not a good use of resources to test every person who comes in a clinic, but maybe reserve that for people that are either having a lot of side effects to every medication or are simply not responding to conventional treatments. But otherwise, that has limited utility. And these genetic mutations may predispose a person to developing a psychiatric illness, but there is really no single gene that will, if you have that gene, you will get that disorder. The closest thing we have to that, which is kind of a psychiatry adjacent disease, would be Huntington's disease, which is usually treated more by neurology, but people can have psychosis-like symptoms with Huntington's disease. That is a single gene illness. So if you have that gene, or if you have an identical twin who has Huntington's disease, you will have that illness. But most of our psychiatric illnesses, they are associated with these sort of constellations of genes, but there is no single gene that predicts for them. And one of the ways that we can figure this out is by looking at identical twins who have the same DNA. But even a disease like schizophrenia, the concordance rate, which is the likelihood that if one twin has the disease, the other twin will also have it, is only about 50%. in schizophrenia. And so what that tells you is that there is a strong genetic component, because 50% is a lot, but that there's still this other component which is largely environmentally determined. So we've got a long way to go in understanding how the genes associated with these mental illnesses uh, predicts the course of the expression of that illness phenotype. Now, a lot of times in this class, we're going to be talking about receptors and neurotransmitters. And so these characterized up here, and we'll be unveiling these characters as we go through this week and next week. So these receptors, they're a specific molecule, they're a protein that sits on the surface of the neuron and extends into the inside of the neuron. So these are known as transmembrane. So they're sitting on the cell membrane, or they can be inside the cell. They can be intracellular receptors. And these receptors are attached to by what are known as ligands, or sometimes in some parts of the country, they refer to them as ligands. But these are neurotransmitters. So neurotransmitters are ligands. These ligands will attach to the receptor and they will activate that receptor. Now, drugs can also activate these receptors. So our neurotransmitters are what we call endogenous. They come from inside of us. Drugs come from the outside. They are exogenous. And depending on the shape of that drug and the properties of that drug, it may activate the receptor in the same way that a neurotransmitter does, or it may block that receptor, or it may stop that receptor from activating at all. And when we hit these receptors in the brain areas that we want to target, that gives us the target effects. When these receptors are activated or blocked in other parts of the central nervous system or other parts of the body where we don't want it, that's where we get side effects. And we'll come back to that time and time again. So let's go back to cell biology 101. This is your classic diagram of a presynaptic neuron. You can think of that as the pitcher, and the postsynaptic neuron, which you can think of as the catcher. So when this signal comes down the line, the cell fires, neurotransmitters are released from these ready-made packets called vesicles. They float across the synapse because remember, the neurons do not touch each other directly. They are separated by these microscopic gaps called synapses. And on the other side of the synapse is that catcher's mitt, or that receptor. And they are just waiting there to receive those that neurotransmission in the form of a receptor. So they bind to that receptor on the postsynaptic side. So the releasing neuron is the presynaptic, the catching is the postsynaptic. Get used to that terminology because we'll use that a lot. And then once it activates that receptor on the postsynaptic side, there's some kind of response that happens. And that can be a slow response or it can be a fast response. And we'll talk about some of the differences with that later on in this talk. And then the signal is passed down the line. Now, these neurotransmitters will fall off the receptor and they might just diffuse away, or they might be picked up by the reuptake pump there. You can see that's the serotonin transporter. It could be a norepinephrine or a dopamine transporter. So it's pulling that molecule back into the presynaptic side, where either one of two things happens. One, it can be broken down by a degrading enzyme like monoamine oxidase, which reside on the mitochondria in the presynaptic neuron, or it can be packaged back up. Seeing that little, that green one that's spinning right there, that would be a vesicular monoamine transporter. So VMAT is what that's abbreviated as. And its job is to repackage that recycled neurotransmitter and get it ready to be used again. This is an efficiency mechanism on the part of the body. Rather than having to make this neurotransmitter from scratch, it essentially recycles the old material, which is an efficiency mechanism. So let's start with ligand-gated ion channels, which are also known as ionotropic receptors. So conceptually here, what we've got is essentially like a tube or a funnel, if you will, that sits on the surface of the membrane and pokes through the membrane into the intracellular space. When that when that opening is activated by a neurotransmitter, it allows for ions, which, if you remember, are positively or negatively charged molecules. So positively charged ions are cations, negatively charged ions are anions. So those will float through that tube that the receptor is comprised of into the intracellular space. And depending on if those are positively or negatively charged ions, it's going to have some influence on whether or not that cell fires. So a good example of this would be something like GABA receptors, which are involved in allowing negatively charged ions in to inhibit the firing of a cell. And we'll dive deeper into that in a moment. So these are fast acting. These kind of receptors work very quickly, which is why, for example, why benzodiazepines and alcohol do not take weeks to work. They happen quite quick. They have an effect very quickly because they are influencing these ionotropic receptors. So depending on the net amount of positive or negatively charged ions flowing into the cell, that will either tip the cell away from firing if it becomes more negatively charged, or it will go towards firing if it becomes more positively charged. So going back to Physiology 101, you probably remember that the inside of the cell is more negatively charged than the outside of the cell. So really, you've got these sort of positively charged ions outside the cell, kind of waiting to come in. They're like waiting to rush into an department store on Black Friday, if you will. So they're going to go down the concentration gradient if they're given a chance. They'll go from an area of higher concentration to an area of lesser concentration. So when we're at rest, we see this negatively charged respective to the outside resting state. So usually about -70, -80 millivolts. If the cell becomes more negatively charged, we call this hyperpolarization. So it goes even further away from the possibility of firing. As positive ions flow in, it moves towards depolarization. And eventually, if the cell fires, the entire cell will depolarize and then repolarize. And how does it maintain this resting negative state? If we again go back to our Physiology 101, remember we have these sodium potassium pumps, and 3 sodium ions are pumped out of the cell for every 2 that are brought in. So what that gives you is a net negative charge. It's kind of like we're cocking the hammer on the gun, if you will. We're getting this thing ready to fire. As the cell begins to depolarize, so as this charge comes down the membrane, the sodium begins to flow into the cell. So these, remember, sodium is Na plus. It's a positively charged ion, comes through these sodium channels, and then eventually, as this action potential occurs, those sodium channels close and potassium flows in, which allows the cell to fully depolarize. And then those sodium potassium pumps go back to resetting the trigger, if you will, each time after the cell has fired. But once the cell begins firing, it goes all the way through. So it's kind of like, you can think of it like when you flush your toilet. You can't stop the flush of a toilet midway through, right? Once you push that lever down, this entire cycle has to happen before the toilet can be flushed again. And similarly, a cell that is fired, there's this refractory period where it can't fire again for a very brief period of time while it is recharging. Silly science nerd joke here. So these ionotropic receptors, they allow these either positively, depending on which receptor they are, they either allow positive or negatively charged ions to enter inside the cell from outside the cell. So there's more of those positive ions outside the cell. It's like all the people waiting for the sale on the morning of Black Friday. As soon as that door opens, as soon as think of that ligand as the person who unlocks the door and opens it up, what's going to happen is that pore, that manhole, if you will, it's going to open up, and it's going to allow those ions to flow inside. Okay, so that's going to go down the concentration gradient. There's higher concentration outside the cell than inside the cell. And if enough positive ions flow in, it will cause it to fire. So now let's take a look at some of these examples, some of these positive, these ionotropic receptors that allow these uh, positive ions to flow in, which is going to move the cell more towards excitement, more towards depolarization. So example, these would be glutamate receptors, very important in the use of, say, something like ketamine. Serotonin 3 receptors, which are common on the areas of the brainstem, the chemoreceptor trigger zone, which are responsible for nausea and vomiting. So sometimes when we give somebody a drug that increases serotonin, like an SSRI, we actually, that extra serotonin activates those receptors as well, which may be one of the reasons why we get nausea. That receptor, by the way, is also a receptor that we block with the drug ondansetron or Zofran. So as when that opens up, these positively charged ions flow in, and the cell moves towards depolarization and an action potential firing. Now, GABA and glutamate are really the main players in the brain. You know, in psychiatry, you think we talk a lot about serotonin, we think about dopamine, but really GABA and glutamate are doing the vast majority of the work. Over probably 80% of the work. And we can think of these things as being widely distributed in the brain. So most every cell type neuron has GABA receptors, and it also has glutamate receptors. And these are going to have an impact on whether or not that cell fires. So let's first take a look at that excited fellow there. That's going to be glutamate. So glutamate is the most common excitatory neurotransmitter. And we can think of this as kind of the floor manager. That's like, okay, speed it up. We can also think of it as like the gas pedal in the brain. And there are a couple different types of glutamate receptors. So there are those ionotropic glutamate receptors that we already talked about. These are the ones that allow these positively charged ions, usually sodium or calcium, to flow in, which allows that cell to move towards depolarization. That's that speeding it up, moving it towards an action potential. It's an excitatory action. Additionally, there's another type of glutamate receptors known as metabotropic glutamate receptors. These are slower acting. These are not ion gated. These are not these ionotropic kind of channels. These are something called G protein coupled receptors. And we'll talk more about those in a moment. But those are slower acting kinds of receptors. So the ionotropic glutamate receptors would be things like NMDA or AMPA or the kainate receptor. And then there are 8 different types of these, what are sometimes referred to as mGlu, which means metabotropic glutamate receptors. Those are those slow acting ones. Now, the interesting thing about glutamate receptors is, so we've got those you see these here in the on the diagram, these green and these orange ones. These are these fast acting. These allow this little pore to open up. Now these positively charged ions can flow in. It's going to make this cell more likely to fire. Now, what's interesting about this is that you'll notice there's this astrocyte here. Now, we talked a little bit in last week about astrocytes being part of the blood-brain barrier. Astrocytes also do another role. So these are glial cells. They exist right next to neurons. And part of their job is to process the used glutamate, if you will. So instead of having a reuptake pump on the presynaptic terminal, this is actually moved through astrocytes and brought back into, is recycled through the astrocyte back to the presynaptic neuron. I also want to point out those slow acting metabotropic glutamate receptors. So these are those G protein coupled receptors, slower acting. They work differently than these ionotropic receptors that we've talked about here. I also want to point out that these AMPA receptors and NMDA receptors, not to be mistaken with MDMA, I know it sounds pretty similar, but NMDA, which is the glutamate receptors. So I want you to watch this. I'm going to play this a couple of times. So watch that little blue star. That's a molecule of glutamate. And you're going to notice what it's going to do is it's going to lock onto both of these receptors, both the AMPA and the NMDA. And then I want you to watch the AMPA one first, because we're going to break this down here. So here comes the glutamate. These open up. See that little funnel open up? And the sodium goes rushing in, and the potassium goes out. Okay, now I want you to watch the second half of this, so the right half. So let's go back. Okay, so this time we're going to watch the glutamate attached to both of them, but this time I want you to watch the NMDA receptor. And the things I want you to watch for is when that opens up, you're going to see a little lightning bolt appear next to the NMDA receptor. And what that is showing us is that the AMPA receptor has opened up. It started to allow these sodium ions to flow in. The cell is beginning to depolarize, and it's actually sending that depolarization down the cell membrane. When it hits that NMDA receptor, that depolarization is going to kick a single molecule of magnesium out of that cell channel. You'll see that pop out. And when that happens, the sodium can flow in. So let's go ahead and watch all that. Here comes the glutamate, goes the AMPA, cell fires next to it, kicks that magnesium out, sodium flows in, calcium flows in, the cell begins to depolarize. And this will happen all the way down the membrane of the cell, carrying that depolarization or that action potential forward. So it's a sort of back, forth, as it goes down the length of the cell membrane. So drugs that affect glutamate. So some you may already be familiar with, such as ketamine or S-ketamine. Also dextromethorphan, which is commonly thought of as a cough suppressant, which it is, also is a glutamate receptor antagonist. The problem is it wears off fairly quickly. So cleverly, somebody thought to add a cytochrome P450 2D6 inhibitor, bupropion, to that combination, because dextromethorphan is broken down through cytochrome P450 2D6. So by using bupropion to inhibit that enzymatic pathway, the dextromethorphan sticks around longer, and it can exert this antidepressant effect, somewhat like ketamine. But this can be taken on a daily basis and should have an effect on depression fairly quickly. Another drug, S-methadone, which is the left side of the racemic methadone mixture. So methadone, which we think of as an opioid agonist. The left side, the left enantiomer, S-methadone, actually works primarily on glutamate. There are also drugs that prevent the release of glutamate. So lamotrigine would be a good example of that. There are also things such as N-acetylcysteine, which help to reduce the amount of extracellular glutamate, because you don't want to have too much glutamate. It becomes what's known as excitotoxic. So N-acetylcysteine is commonly used in a number of different situations. If you've ever given mucomist for somebody who has taken an overdose of acetaminophen, that is N-acetylcysteine. We sometimes use N-acetylcysteine in the treatment of things like OCD, addictive disorders, and other kind of compulsive behaviors. There's also been some interest in looking at metabotropic glutamate receptors as a possible antipsychotic action. That's still very much under investigation. There are no commercial drugs available that do that right now. So let's shift our attention to the other side of the coin, these ionotropic receptors that allow in the negatively charged ions. In this case, we're mostly talking about chloride ions. These are chloride anions, they're negatively charged. And the best known one of this would be GABA. So GABA, of course, is the primary inhibitory neurotransmitter of the brain. So when GABA, the neurotransmitter, which is symbolized by that little orange plus there, comes and locks onto the receptor, you see what happens? It opens up, allows these negatively charged chloride ions to go down the concentration gradient. There's more of them outside the cell than inside the cell. They flow in, and they decrease the probability that that cell is going to fire. So they make it hyperpolarized or more negatively charged, which is going to inhibit that neuron from firing. So you can kind of start to see where this is going, right? So when we think about something like a benzodiazepine for anxiety, what are we doing? We're slowing down neural activity. So we can think of GABA as kind of the union stewards. They're like, hey, slow it down. Don't make glutamate make you work too fast, right? Primary inhibition. And they are found, GABA is found all throughout the brain. So just like there are glutamate receptors on all neurons, there are also these GABA receptors on other neurons. And most of this is being secreted by what are called local interneurons. So we can think of GABA and glutamate as being sort of widely distributed throughout the brain. Some of the monoamines, as we'll talk about later on, they come from very localized areas of the brain, and they have to come a long way to get there. So you can think of those as almost like, if you've ever ordered anything from Temu, I think it comes all the way from China. You order it, you got to forget about it, and it shows up 4 weeks later, and you think, oh, what's this package, right? Whereas GABA and glutamate are more like Amazon. So Amazon has these distribution centers everywhere. So this is why you can get your goods in a few hours or maybe a day, because the things that you need are probably in a warehouse not too far away from where you live. And so similarly, GABA and glutamate are found all throughout the brain, so they can get to where they need to right away. So GABA, the neurotransmitter, when it activates these ionotropic GABA receptors, it's going to allow that ion channel to open. Those chloride ions are going to flow in, and it's going to make it less likely for that neuron to fire. So we can think of that as kind of the brake pedal of the brain. And of course, with the brake pedal, we can tap it very gently, slow things down a little bit, and that might be like the first drink you have at a cocktail party. If you have two more drinks, you're pushing a little harder, right? Now maybe you're losing a little bit of coordination because you're inhibiting those pathways that correct for balance and such. If you have a couple more drinks, maybe you get sleepy. We're suppressing neural activity in general. If you take a couple more drinks, you might fall asleep. And if you took 5 more drinks, you might stop breathing, right? So you're pushing the brake all the way to the floor at that point. So in a dose-dependent way, we can influence the amount of GABA activity that's happening in the brain. And this, of course, is what's responsible for, if somebody overdoses, say, on a barbiturate, it is these negatively charged ions flowing into the cell and inhibiting vital activities such as respiration. So let's take a look at these GABA receptors. So as you can see from this red line here, they are transmembrane receptors. So one side sits outside the cell, the other receiving end, the bottom end, sits inside the cell. And that blue funnel-shaped thing is that central pore, that thing that's going to allow those negatively charged ions to flow in. If we were to look at it from the top, you can kind of see it like a manhole, if you will. So once this opens up, it allows those negatively charged chloride ions that are just sort of waiting for their chance to flow in, and they'll flow into the cell and decrease the probability of it firing. And this, of course, can happen really fast. This is why, say, a benzodiazepine works in the course of just maybe a few minutes to an hour. And as you see, the receptor, the outside of the receptor, is peppered with all these different receptors. We've got our endogenous GABA receptors, but we also have sites that attach for alcohol, for the benzodiazepines, for the Z drugs, if you will, for barbiturates, some of the general anesthetics, and all these will influence whether or not this opens up further in the presence of endogenous GABA. And these, as I mentioned before, there are these interneurons, which really are kind of woven between other neurons, and they synapse onto these GABA interneurons will synapse onto them and express these GABA. They'll release GABA onto GABA receptors on the other neuron and sort of say, hey, quiet things down a little bit. So this is part of the inhibition process in the brain. I want to speak for a minute about allosteric modulation and orthosteric modulation. So if you think about this, so what's happening here when we think about, say, a benzodiazepine, you'll notice many of them end with the letters PAM, PAM. It stands for positive allosteric modulator. So what that means is when if some receptors will have these two additional sites, an orthosteric and an allosteric site on them. When you attach a positive allosteric modulator and then you bring in the endogenous GABA, it's going to cause that receptor rather to be more, it's going to have a greater response. So when we add a benzodiazepine in the mix, it's a positive allosteric modulator, and then GABA comes along, that ionotropic receptor is going to open up even more negatively charged chloride ions are going to flow in. It's going to prevent that cell from firing. So drugs that are relevant to GABA would be things such as alcohol, which works on those alcohol sites of the ethanol sites of the GABA receptor, benzodiazepines, which many people are familiar with for both anxiety and sleep, the Z drugs like zolpidem for insomnia, the barbiturates, which are less used in psychiatry now because of safety issues, but are still used in treatment of seizure disorders. My dog actually takes phenobarbital to prevent his seizures. And then recently we have a non-benzodiazepine drug that works on GABA receptors called allopregnanolone, which is marketed as the drug zuranolone or brexanolone, which is a treatment for postpartum depression, which we'll get into later. I'm going to go ahead and take a quick break here, and then we're going to move on to the slower acting receptors in the next part. So stay tuned.